Welcome to ST World Affairs. We got two executives from Labcyte Corporations, Rich Ellson and Mark Fischer. Mark became president and CEO of Labcyte in September 2008. He has more than 30 years of experience in launching novel diagnostic, medical devices, therapeutic, and high technology growth companies, building from startup through IPO and into high revenue levels. Earlier in his career, Mark held several positions, including vice president, finance, corporate controllers, and advanced technology business managers at a manufacturer of disk drivers that grew from a startup to $1.2 billion in revenue and also went public. Mark holds a BA degree from Stanford University and MBA in marketing and finance from University of California at Berkeley. My second guest, Richard Ellson. Rich is a founder and CTO of Labcyte. Before founding Labcyte in 2000, Rich was area manager at the Xerox Palo Alto Research Center. Rich is inventor of more than 60 US and international patents. Rich holds BS in fluid and thermal science and an MS in mechanical engineering from Case Western Reserve University. Mark, Rich, welcome to ST World Affairs.
Thank you.
Thanks for having us.
Mark, let me start with you. Some of our viewers are not aware of Labcyte. So what exactly Labcyte does?
Labcyte has the ability to provide instrumentation software and automation that incorporates incredibly novel technology that Rich and his team invented, which is using acoustic energy to move liquids with extreme precision and accuracy. As a result of that, it's had a major impact on drug discovery, diagnostics, and a number of newer areas in the world of genomics. So it's proven to be a versatile tool that's had a big impact on new discoveries.
Okay. Sounds like a disruptive technology. Rich, can you go over briefly about how acoustic technology works?
Sure. We can have some video too to show you some of the concepts that are key to how we move liquids. The most important concept is that we can move liquids without touching them. As Mark said, we can take acoustic energy and we use that to make a very small drop. We can propel that drop, and typically we'll propel that upward so that it can actually stick to other surfaces or actually merge in with other liquids. So by making a large number of these drops flow out from our system with acoustic energy, we can transfer liquids to assemble assays and to do important experiments for research in the life sciences. Typically, our customers would have a wide variety of different liquids, and they would store these in what's called microplates. You can see some of the microplates here in the video can hold, from hundreds to sometimes even thousands of different materials. And really where the instrument excels, as Mark said, is with software and automation, allowing you to program how you would build complicated experiments by loading up all these materials. These materials come in on plates that can be loaded by robots and entering into the instrument that you see here, the Echo 550, which is our sort of flagship instrument that we use to launch the technology. So these plates will come into the instrument, again, either by hand or with a robot. And because those little droplets that you saw are so small, in fact, it takes 400 million of them to fill up a 1-liter bottle, that if you actually start to throw them around and there's static electricity, other charge that comes from moving them with the robots, you can actually deionize them. Once they're inside the instrument, we'll actually have a little conversation with the liquids. We'll send sound energy up and we'll bounce that sound energy off the surfaces that make up those microplates as well as the liquid that's in each well. And we'll listen to those signals. And a lot of complicated signal processing goes on to understand the nature of the liquids. In particular, well, are there any in the well? Is the well empty? How deep is the liquid? We'll also use medical ultrasound-like technologies to understand the nature of the liquids that are in the wells. From that information, we can figure out how to actually send the sound in to create drops. There's also some complicated technical issues in moving around liquids, because suppose it's honey or water. Well, it might take more energy to move something that's very sticky like honey. So we also have a conversation with liquids that are potentially very sticky to make sure that we put in enough energy to launch those as well. So they'll fly up and stick to the container that's overhead, as you can see here. So the liquids that could be, in this case, the blue or yellow, would then be shot up into the upper wells and in rapid succession. And the reason this has become so critical in many of the areas that Mark had talked about earlier is the fact that we can send a droplet up out of one well, send it off to a receiver But now we can move that acoustic generator to another well. We can also move the plate that's on top to bring another destination up above. So we can mix and match all these drops in an infinite number of combinations. Really whatever it is that the user wants, they can extract any amount they want from a source and send it to any destination to create very complicated assays. And this is really critical in doing assays and experiments, particularly in genetics and genomics, where you may want to pool lots of materials together to do experiments inexpensively.
Right, so some of our young viewers who might be watching this video, they might be wondering, do you have to go against the gravity? I mean, do you have to shoot upside down? Why can't you just collect the liquid?
Yeah.
I mean, that's something, it's kind of a valid question. I understand that there's a microplates, you have it, right? And then you put a transducer underneath it. Why can't you just work with the gravity and do exactly opposite what you're doing?
Well, one thing your younger viewers may know if they've watched movies or some of these animations like ants is that surface tension is very different when you're small. If you're an ant, you don't really need a drinking cup, right? You can actually have coffee or your juice on a lollipop, because if you dip a stick in to your cup, you can pull the liquid out and it will just form a little ball on the end of the stick. So surface tension is really, really powerful if you're really, really small. Gravity ceases to be important and surface tension wins. And we take advantage of that in our instruments to have the relatively large volumes that we have in the plates that are at the bottom be the source of the liquid that then flows upwards and sticks to the, essentially by surface tension, upside down. So for ants, this makes a lot of sense. For people, we have to think about it a little bit.
Okay, sounds good. So now that we understand Labcyte technology, Mark, can you walk us through what are the Labcyte product pipelines existing and the future? And then in your answer, you can explain to us in which particular sectors or area Labcyte are dominating this technology.
Yeah, I'll start with the last part first, and that is we started off in the world of drug discovery, supporting high-throughput screening in all 20 of the top 20 pharmaceutical firms around the world. And as a result of that, this technology is now foundational to the ability to investigate new compounds that might become a therapeutic, which is an essential operation for pharmaceutical firms to start their activities. In about 4 or 5 years ago, we were able to take inventions from Rich and his team to be able to automatically transfer any fluid anywhere, anytime, whether it's very viscous like honey or whether it's another type of solution. That enabled us to get into the world of genomics and proteomics, including into synthetic biology and diagnostic areas. And with that in hand, we currently are able to support literally hundreds and hundreds of applications with the ability to miniaturize the transfer of the samples, which in turn is a massive cost reduction and a large improvement in data quality. And that's what really needs to happen to provide an engine for the major advances in healthcare that everyone's looking for. So we're well poised and primed to be able to support in a whole slew of areas, whether it's drug discovery through to diagnostics. All areas of life sciences involve moving liquids, and we do it in a way that can really take advantage of the newer tools around detection capabilities that are currently, in a way, bottlenecked by decades-old technology. Mm-hmm. So we offer the automation systems and the instruments that do the liquid.
That's your product pipeline.
As our product pipeline. Right. And we're in an environment where we continue to support researchers doing brand new application areas, including in personalized medicine. And 16 researchers in the Institute of Molecular Medicine in Helsinki, Finland, figured out how to use our Labcyte system to do the following. Take patient cells from people who are suffering from leukemia. They're literally days away from passing away. And the clinician is asking, well, what drug do I give to this patient? By using the Labcyte system to transfer droplets to be able to see, well, what cancer therapy that's on the market today might work on that particular patient. So if the cancer is killed in the microtiter plate well, that has a much better shot of being effective than if it's not. Mm-hmm. And as a direct consequence of that and collecting genomic information, you put those together and you have really now true personalized medicine that enables then the clinician to have a better shot at a drug that will work for that patient. As a direct consequence of that, patients have been put into remission, and there's been a drug for kidney cancer that now looks like it works really well for leukemia. So that's a major advance, and this modality is now being used more broadly in Europe.
So let me ask you a follow-up question. When you look at the compounded annual growth, compounded CAGR for pharmaceutical is like roughly like 4 or 5%. So it looks like Labcyte future strategy or future growth is going to be in genomics and science, and that's where I think Labcyte will be mainly focusing on in coming years. Is that the focus? Is that the strategy Labcyte has it?
Yeah, in our envisionment of the world, in the near term, there's going to be continued growth in the genomics area, and this includes synthetic biology applications where people use the echo to stitch together DNA and gene constructs of interest to put into organisms that in turn makes things like penicillin or biofuels or other things that people want to get, flavors and fragrances, for example. And the Labcyte ecosystem is very popular for doing that work because of its accuracy, precision, and miniaturization capabilities. And so we're going to be growing in the near term around genomics applications. And then we have very exciting products coming out next year that take advantage of very strong diagnostic tools, or analytical tools rather, of mass spectrometry, and interface the unique features of acoustic droplet ejection with mass spec systems to be able to have a potentially dramatic impact on drug discovery, synthetic biology, and other areas. Okay. So we have other products that are in the hopper, as well as growing off of our base business that we have today.
So this is fantastic. So Rich, so can you walk us through your experience going from those early days, and how did you come to this point?
Yeah, absolutely. Traditional liquid handling technologies are things you may have seen, your own child do at a table in a restaurant, much to your dismay, is to pick up a beverage, right, in a straw, put their thumb on the top, carry it over, and release it. And believe it or not, that's how most liquid handling is done if you don't use an echo around the world today.
That's
essentially people
pipetting,
like a pipette tip, right? They'll suck up fluid, right? And they'll hold on to it, and they'll move it somewhere, and then they'll release it. And that technology has had a lot of problems. In particular, the smaller you make the straw, the harder it is to control the amount that you release. So a lot of the advantages that we've had from our technology, as Mark said, was that precision and accuracy that allowed us to make everything smaller, because we're not touching it. We can actually control the amount with the technologies that we showed earlier in the video. And so to move away from a drinking straw, which is how people had moved liquids in this arena for decades, right, if not longer, centuries, was very disruptive, and it was very difficult for people to wrap their minds around the fact that we could move things without actually touching them. And quite honestly, in order to raise money, we literally had to bring people to the garage. Yes, we started in a garage here in Silicon Valley.
Just like typical Silicon Valley startups.
Yep, and we said, well, you don't have to believe us, just come look at it. And once they saw it, of course, it was very persuasive that the technology, would work. Of course, there were still a lot of barriers. Just making little droplets like is on the video doesn't mean you can make an instrument that works with robotics and with the range of materials that people want. That obviously took a lot of hard work from a lot of people to make a box that could deliver that reliably. But really, the initial persuasion was, seeing is believing, and showing them that focus sound could be a controlled method to consistently deliver fluids. And that was probably our biggest obstacle, was to create that first prototype and make it visible to people, because it is very little, so that they could actually see the process and how stable it would be in a prototype phase, so that it could move and to get the resources to move it into commercial production.
Okay. So now fast forward, after 14 years, Labcyte is here. So can you just walk us through an example of single cell sequencing? How does it work? How does Labcyte instrument and all the automations, and how does it help in a performance point of view compared to traditional?
Well, single-cell sequencing is a great example, because here was a technology that was sort of stuck. People realized that they only wanted to have one cell, and if they want to amplify the DNA in one cell, they needed to start with one, and they also needed to be able to replicate that in such a way that they could recover it, concentrations that can then be loaded into modern sequencing devices. And one of the things that people had learned is the smaller you made the volume and the more viscous, right, more like honey you made the fluid that it was in, the better off the results would be. They could get more uniform coverage. It's sort of like if you had a big pool of stuff and you sent it into a photocopier, your book, and it only copied pages that were 9 and 2 and 6 and 15. There were certain pages, you got 19 copies of those and 2 copies of other pages. It wouldn't be very useful if you wanted to distribute it to everyone, and that's what a sequencer is looking for. But as you started making things smaller and smaller, you started getting 5 copies of everything, very uniform coverage and very uniform copies from the polymerase chain reaction, the process that does this. But in order to do that, you need it to be little and you need it to be sticky. Well, enter Labcyte. We do little well, we do sticky well, and we could automate everything. Not only that, once you were done, you could assemble all the pages in the book, right? The process that we have from going to any well is a very efficient method of binding all those pages or stapling them, if you continue with the copier analogy, all into a nice, neat package. And that actually turned out to save people 99% of the reagent cost would go away, and the quality would be better. So it was smaller, faster, better, and they could assemble all that stuff with the automation that we provided, all in one shot. So this was very enabling for people who wanted to understand, well, I'm going to take, say, all the bacteria that's in the gut of a termite, because they digest, interesting, materials, cellulose and other things. People want to understand how that works. But how do you find out what all those little bacteria are if you can't grow them? Well, people don't know how to culture things they don't see. So how do you get enough material from one cell if you can't then grow the cell? So our technology allowed people to solve that problem. They could just take one cell and they could use this small and viscous environment to then replicate the DNA with chemicals rather than with culturing, growing it in a petri dish and scraping it all up when they don't know the recipe for how to make those cells happy. So that was a very important advance to give people a tool to basically study microbiomes. I mean, we're actually made up of more cells that aren't, human origin cells than we are cells that are from people, from all the other things that are living in us. But we didn't have a tool to allow us to pluck one cell at a time, make those uniform number of copies in a chemical process, and then load them into sequencers. And that's been one of the major advances, is really a tool to discover the microbiome. That's come out of echo applications. And that's really been in the last couple of years that our collaborators at the Joint Genome Institute right here near here in Walnut Creek started publishing papers saying that this is now becoming possible. We can actually get good data from one cell and understand these microbiomes. And we've now had quite a few sales in that area, people that are interested in, say, probiotics analysis or understanding the human gut microbiome and even how that can be harnessed to improve human health.
So, Mark, just recently Bill Gates mentioned that AI, artificial intelligence, energy, and then bioscience or biotechnology, those are the 3 fields. It's good for anyone who wants to get into that. You can find a good job for career point of view. So for our young viewers, again, I'm asking this question from a young viewer's point of view, if they want to get into this biotechnology, bioscience, or genomics space, how Labcyte fit into this biotechnology or bioscience? I mean, from technology point of view, and from a growth point of view.
Biology is such an incredibly exciting space and time today. And the ability to have tools now, such as the Labcyte equipment, that really, for the first time, allows scientists to control liquids, which is a rather astounding statement when you think about it, is so impactful that it's going to contribute to these major advances in scientific knowledge. And that includes new therapeutics. It includes the ability to create organisms that do incredibly powerful things in terms of secondary creation of other chemicals. And in that environment, boy, what a fun place to be, where you're building and growing and constructing things with accuracy that you didn't have that tool before. With respect to artificial intelligence, for example, we all know that they're going to be mining papers and periodicals and those kinds of things for data sets, and we know a lot of those data are not reproducible. And it's our view that a number of those publications are not reproducible because of errors in liquid handling. And there are many, many, many sources of errors in liquid handling. So if you can obviate that, you have a much better chance of avoiding the problem of artificial intelligence that has garbage in, garbage out problems as a direct consequence of poor liquid handling. So this is a seminal advance that is going to be a very, very powerful, and for people looking at the world of biology, right now the ability to advance through the creation of organisms is going to be incredible in terms of its impact.
Okay. So in terms of your future product line, there was an announcement and all over the places about this acoustic tube. Can you just share us some more information about that?
Sure. One of our, if you step back a little bit and look at the big picture within the world of research, and that is an environment where there's been billions of dollars invested in new detection tools and technologies, whether it's mass spectrometry systems or systems from Illumina for next generation sequencing or other modalities. And really what the giant bottleneck is now that everyone is realizing is the ability to manipulate and manage sample preparation and liquid handling to feed into those now very sophisticated, highly specific detection systems. So we, Labcyte, have that capability now to revolutionize that whole space with our miniaturization and control over liquids, and that's what we're doing. The front end of that process, though, is how do you get samples or materials, into that preparation stage. And to accommodate that, Labcyte has also developed a tube container in addition to the microtiter plates that are acoustically friendly, that in turn can store not just compounds, but also biological fluids. And in that context now, this becomes the first step in handling of the liquid from a sample perspective, and then processing a liquid and the associated chemistry used in detection for the Labcyte ecosystem to play in that entire space from A to Z. So pharma, for example, is one pharma group is literally blowing up their entire infrastructure around how they store and manage their 4 million compounds that they have in storage for evaluation, and they're going along with an acoustic tube product that is being developed by Labcyte. And then, of course, this will all be used in biostorage as well.
Okay. All three of those areas sort of play together, right? The ability to process data and to do the data mining from all these data sets. One will be cleaned up. Well, actually, we have people now rerunning experiments to really understand, what was the actual result, because it's sometimes with other processes interfering, like having tips that touch things and don't give you the right transfer amount, or things that stick to them, they would get bad data. So a lot of that cleanup is going on, but also the ability to mine more data. In the genomics area now, if people can actually do 99 times as much work, right, because they've shrunk all their processes down using the echo, they can generate a lot more data. So instead of, say, just sequencing one person, you could now, say, sequence cells that you would take from all over their body and understand an evolutionary tree of how all the cells in their body came from those progenitor cells, and really look at someone like you would their ancestry, but all coming from that one cell propagating through the whole body. So if you can do 99 times as much work, you can start to think big. And this takes a lot of informatics to understand how, say, people would evolve from a developmental biology perspective. And similarly with cancer. Mark had mentioned the work that we're doing with FEM. Though they're looking to try to understand how not just do drug discovery to help a large population with a drug that might work for them because statistically it looks good. They were taking those drugs and looking at how they could work well for an individual. But we can do the same thing at the cellular level with their cancers. We can now not just say, well, typically a cancer like this might have this genomic profile. We can go in and take individual cells from one person's cancer, and again, cost effectively go in and sequence those cancers and understand the evolution of that person's cancer. Okay. So all these technologies started to come together and allowing you to process data, to get information, and eventually to use the results of other studies that you do with populations and the mutations that you find and apply those into drug treatment. So it's really quite a synergistic relationship between all those areas.
Okay, so Mark, let me ask you a question from an entrepreneurship point of view. So far, Labcyte have raised less than $60 million within 5 rounds. Walk us through, again, from layman's term, that what it means to be raising money from seed to series money, and then what is Labcyte future plan? What's the exit strategy?
Sure. So in the traditional Silicon Valley development process, if you will, there's a formation of a company. There's often money that's obtained as seed financing to do some early work in terms of starting the assembly process for that particular company. Then there's funding from typically venture capital firms that would provide the series of stages of funding, and they're known as Series A, B, C, and they go on. And in that environment, the investors that come in to get stock in the company in exchange for cash are looking for advances and milestone indications of where the company has progressed to in order to justify the risk of making a substantial investment in the company. For us, Labcyte, we've been incredibly capital efficient, which helps the risk profile for any future investors coming in related to continuing to grow our company. So far, we've been in mode, since I joined Labcyte, of more, as I call, pay-as-you-go strategy of being able to fund our growth out of our existing resources, along with some working capital debt and some other elements. And we're in a situation where we're getting closer and closer for the ability to do an initial public offering, which means that we would become public, and that means that somebody could buy shares of Labcyte stock in the open market. And that's something that we've achieved a critical mass that would allow us to accomplish that goal, which in turn would get the original investors in the company a return on their investment going forward.
Okay, fantastic.
Yeah.
Rich, Mark, I wish we had more time. Thank you very much for coming down to ST World Affairs. Thank you.
Thank you.
Thanks very much.